Welcome to today's lesson, which is on glycolysis. Last time we took a look at the overview of cellular respiration, and it's important to keep the big picture in mind. So in cellular respiration, organisms take sugar and break it apart to release some of that chemical energy and use it to make ATP, which is what our cells run on. ATP is what you use to run your body, to do the things that you do, think, walk, move your muscles. All those things require ATP. In this process of breaking apart sugar, we need the help of oxygen, and carbon dioxide and water are produced as byproducts. So this is the big picture of cellular respiration. Cellular respiration is actually a summary of all of the many chemical reactions that take place as glucose is broken apart. And here are the three main steps of cellular respiration: glycolysis, the Krebs cycle, and the electron transport chain. And last time we took an overview of these steps, and we know during glycolysis we make 2 ATP, during the Krebs cycle we make another 2 ATP, and during the electron transport chain a whopping 32 to 34 ATP are made. However, the electron transport chain is not possible without getting stuff from glycolysis and getting stuff from the Krebs cycle. So today we're going to be concentrating on this first step of the cellular respiration equation, and that is glycolysis. And during glycolysis, we go from glucose, which is a 6-carbon sugar, and we break it apart into 2 pyruvates, which are 2 3-carbon molecules, and we produce 2 ATP in the process. But before we look at that, we're going to discuss cellular respiration as a redox reaction, because that is what it is. In cellular respiration, our glucose is oxidized into carbon dioxide. It becomes electron poor. It loses its electrons. And oxygen, because redox reactions come in pairs, oxygen becomes reduced as it forms into water. But we're going to focus right here on this part. How do we get from glucose down to carbon dioxide? And what's happening here is a controlled fall of electrons. So as glucose is broken apart, as it is oxidized, you're going to have a controlled fall of electrons. And what happens during the many steps that break apart glucose is that electrons are going to be harnessed and they're going to be captured at each step. And those electrons are going to be captured by things known as electron carriers. All right, so the first electron carrier we're going to discuss is NAD+. So let's do a little review of electron carriers. An electron carrier is a molecule that is capable of accepting electrons from one molecule and donating them to another. As the electrons are transferred, energy is released. You've already seen electron carriers back when we discussed photosynthesis. During the light-dependent reactions, remember, we went from NADP+ to NADPH. NADPH was the reduced version, and it was used to run the dark reactions, the Calvin-Benson cycle. So that was photosynthesis. You have different electron carriers for cellular respiration. And the ones that we're going to see as the main electron carriers are they come in pairs, like oxidized and reduced pairs. So we have NAZ+ and NADH, which is the reduced version, and we have FAD, which is the oxidized version, and FADH2, which is the reduced version. And you can see the pair of redox equations here involving NAD+ and NADH, and FAD and FADH2. So again, we're going to look at just glycolysis today, and we're going to see how we make 2 ATP and how we break apart glucose into 2 pyruvates. So glycolysis is a process that occurs in almost all cells, regardless of whether that's a bacteria, a plant cell, an animal cell. Glycolysis happens. It's an anaerobic process, so it doesn't require oxygen. Oxygen is going to be required later on in cellular respiration. It takes place in the cytoplasm, and there's a net yield of 2 ATP per glucose. And we'll look at why that's called a net yield later on. Okay, the electrons from glucose are going to be transferred to 2 electron carrier NADH molecules, and we're going to produce 2 3-carbon pyruvate molecules, which look like this. Here are 2 pyruvates. So we've got NADH, we make pyruvate, and we also make ATP. So here are the steps involved in glycolysis. So the first steps of glycolysis actually don't make ATP. They actually use up ATP. So these are our ATP requiring steps. You have to invest some energy into glycolysis to end up with a net of ATP at the end. So in the beginning, what happens is we take an ATP, so we need an ATP, and a phosphate group from that ATP is transferred to our glucose. Here's our glucose, which is our sugar, and we add a phosphate group from ATP to that glucose, and we form something known as glucose 6-phosphate. So that's our first step. The second step, we require another ATP, and we're going to take another phosphate group from that ATP, and we're going to make this fructose 1,6-biphosphate. Now this molecule is unstable. It doesn't like existing, so it's going to break apart into two other molecules, and those two other molecules are two PGAL molecules. They're also known as G3P, and you saw these in photosynthesis. So we form two of these PGAL molecules. That's our second step of glycolysis. Next, what happens is now we start our ATP generating phase. We put in 2 ATP, so we're down 2 ATP. We better start making ATP. So in step 3, what happens is we take our 2 PGAL molecules and we're going to attach a phosphate group to them so that we form 2 of these PGA molecules, right? These are PGAs over here. These are the oxidized forms of PGAL. And since we're going from PGAL to PGA now, which is oxidized, we're losing some electrons, and those electrons are going to be used to make NADH. And NADH is something that we want to make. So we're making 2 NADH molecules over here. Okay, so that is our step 3. So step 4, we're going to start with our PGAs, and we are going to have the help of an enzyme, and it's going to transfer a phosphate group from each of these PGAs to ADP. And now we are going to be forming 2 ATPs. Okay, so we're taking a phosphate group from here, and we're going to form 2 ATPs. Now, in that process, we go from PGA to a molecule known as PEP over here. These are intermediate molecules. And now we're really at a net of we've made 0 ATP. We put in 2 ATP and we made 2 ATP. So we're going to have to keep going for this process to be worth it. So in step 5, you're going to have an enzyme transferring a phosphate group from each of these PEP molecules to ADP, and we're going to form 2 more ATPs. So at this point, we have put in 2 ATP and we made 4 ATP. So our net ATP made is 2 ATP. We also made 2 NADH molecules, and we're left with 2 of these pyruvates, which are going to be used later on. So this is our glycolysis process. Again, we made 2 NADH, 2 ATP, and 2 pyruvates, and we are more or less ready for the next step of cellular respiration, which we can go to if oxygen is available, and that step is going to be the Krebs cycle. Okay, beyond glycolysis, so glycolysis begins the sugar pathway. Depending on the cell type, and the environmental conditions, the pyruvate might go to the Krebs cycle, or alternatively, the pyruvate may be used in fermentation, which we are going to discuss later. All right, so that was a summary of glycolysis. I want to finish up by showing you something kind of rewarding. And these are some ants eating sugar crystals. Okay, and it is marginally relevant because during glycolysis, we go from glucose, which is sugar, and we break it down to release 2 ATP. And in that process, we create something known as pyruvate. So that is what's going to be happening inside of these ants as they break apart the sugar that they are eating.